Hi everyone, I'm Sophie Stevens and I would like to welcome you to my presentation on identifying the primary cells adherent to the arterial sheath of patients undergoing left heart catheterization. Our lab is interested in vascular biology. Shown here is an artery. Blood flows here through the lumen, and radiating outward there are three layers. First, there's a single layer of endothelial cells, which make up the endothelium. Next, there are several layers of smooth muscle cells, and then lastly, there's the adventitia. We study endothelial cell metabolism due to endothelial cells' importance in maintaining cardiovascular homeostasis. Endothelial cells are often studied by culturing commercially obtained immortalized cells. However, studying primarily obtained endothelial cells might provide a better insight into vascular biology because primary cells are exposed to the hemodynamic and metabolic stresses of the body. We have previously studied and obtained endothelial cells from adult and old otherwise healthy men. We first insert a catheter into the radial artery, then we insert a J-wire and obtain a biopsy of the endothelium. We then remove the J-wire and collect cells that adhere to it. Subsequently, we aliquot the cells into an 8-well plate, verify endothelial cells, and quantify proteins of interest via immunofluorescence, which is shown on the right. The bottom left is VE cadherin, which is specific to endothelial cells. The bottom right is DAPI, which is specific to the nuclei. The top left is phosphoinos, which precipitates nitric oxide generation, and the top right is a merge of all three of these. This method of treating and obtaining cells provides many valuable insights. However, it is possible that these quantifications are influenced by other cell types which may be present in the admixture. As you can see from this image taken at a lower magnification, in addition to there being VE cadherin and DAPI co-stained endothelial cells, there are also many other cells seen as DAPI-stained intact nuclei. It is quite possible and likely that these other cells can influence endothelial cell metabolism and therefore the response to our treatment. So, we set out to determine what other cells are present in the admixture in order to determine a protocol to obtain pure endothelial cells. We did this with a different patient population. Instead of using healthy patients from the J-wire procedure, we obtained primary endothelial cells from individuals with cardiovascular disease who were referred for a left heart catheterization. To perform this catheterization, a sheath is placed into the radial artery, which allows the catheter and guide wire to be inserted. The sheath comes in contact with the inner lining of the artery, including endothelial cells and other cells. When the sheath is removed, some of the cells remain. We perform the following procedure to obtain the endothelial cells from this admixture. 5 days a week, on the morning of the procedure, we deliver a cooler with 4 conical tubes filled with dissociation buffer and ice to the cath lab in the University of Utah Hospital. Patients referred for an arterial catheterization for various reasons consent to the collection of the admixture of cells from the catheter sheath that would otherwise be discarded after the completion of the procedure. 0 to 4 patients consent to this on any given collection day. Immediately following the procedure in the OR, the sheath is placed in a conical tube with dissociation buffer and ice and transported back to our lab. The admixture of cells is washed off into the tube, centrifuged, and the pellet is treated with red blood cell lysis solution. The sample is then centrifuged a second time and resuspended in cell staining buffer and incubated in a cell sorting tube with fluorescent conjugated antibodies for specific cellular components. Following the incubation, the cells are centrifuged and the pellet is resuspended and taken to be sorted. Fluorescent activated cell sorting provides a means to determine the cellular admixture with fluorescent conjugated antibodies. We use the following gating strategies to sort. Each thought is first shown enlarged on the right and then becomes part of the figure on the left. First, we use forward scatter versus side scatter to represent size. We then select the particles that are large enough to be considered cells. From this population, we distinguish single cells by gating out doublets, which are 2 or more cells that are stuck together. We do this in plots 2 and 3, which are forward scatter versus side scatter versus area. We then select the live cells with forward scatter versus DAPI. We select the cells with DAPI localized to the nucleus as opposed to cells with DAPI in the cytosol. We then sort the single live cells into CD31 positive and CD45 positive, which correspond to endothelial cells and leukocytes respectively. The CD31 positive cells are then sorted into CD42B negative, which are endothelial cells, and CD42B positive, which are platelets. We then further gate off the CD45 positive cells into CD3 positive, which are T cells, and CD11B positive, which are macrophages. We then further gate off the macrophages into CD14 positive, which are monocytes, and CD15 positive, which are granulocytes. To date, we have collected 36 patients with the previously shown gating strategy. From these 36 patients, we have obtained a total of more than 3 million live single cells, and we have obtained the following average compositions of the cell admixture. This is shown in the pie chart on the left. The cell type and the percent composition are indicated below the chart. There is also an alternative representation of the same data in the histogram on the right. The histogram shows the average number of each cell per sample. Note, there are approximately 200 endothelial cells and 40,000 leukocytes per patient. Now that we know the cell admixture composition, we have two questions. Does the composition change with patients of different characteristics? And two, is the endothelial cell fraction pure? First, we wondered if the admixture composition varied based on patient characteristics such as sex, age, BMI, and diabetic status. To answer this question, patient histories were obtained in a post hoc manner with their MRNs and the medical database. For the purpose of this presentation, I'll be focusing on patient BMI. To investigate the effects BMI had on cell admixture composition, patients were stratified into normal, overweight, and obese categories based off the CDC's BMI classifications. Shown on the left are the average cell compositions of the three categories. As you can see from the histogram on the right, there was no change in leukocytes, T cells, macrophages, monocytes, and granulocytes for any of the three categories. However, there does seem to be a decrease in platelets and also a decrease in endothelial cells for overweight versus obese, as well as normal versus obese. This indicates that obese patients have fewer endothelial cells adhere to the catheter sheath. With the greater N, we can see if this trend continues and becomes significant. Although our data is ongoing, it appears that obese patients have fewer endothelial cells, whereas other cell components are similar. Returning to our original intention of our experiment, we wanted to determine if we obtained pure endothelial cells from FACS. To do this, we froze CD45 positive and CD31 positive fractions, and in a batch analysis, combined 34 patients. In total, we had about 66,000 CD31 positive cells and 1.5 million CD45 positive cells. We then took these cells to the Huntsman's molecular diagnostic core for RNA extraction. We performed a quantitative PCR and extracted the RNA. We also assessed the amount of 4 separate genes. PCAM and the Van Wallgren factor were used as endothelial cell markers, and PTPRC was used as a leukocyte marker. We also assessed and normalized the 18S levels. We observed higher levels of PCAM in the endothelial fraction and very low amounts in the leukocyte fraction, which was used as a negative control. The same is true for the Van Wallgren factor. We saw large amounts in the endothelial cell fraction and very low amounts in the leukocyte fraction. For PTPRC, we saw the opposite. There were large amounts in the leukocyte fraction and negligible amounts in the endothelial cell fraction. Thus, we can move forward with our experiments with primary endothelial cells from patients via arterial sheath and be confident that we are using a pure fraction. I would like to thank everyone in the Simons lab, as well as our collaborators at the U of U Cath Lab, the Flow Core, the Molecular Diagnostics Core, the Cell Imaging Core, as well as Liz Drano. If you have any questions, feel free to reach out.